Good evening, students. Today we are going to discuss about the integumentary system of mammals. So last week I have already completed the integumentary system of birds. So I think you have already gone through the video that I have provided to you. So basically the integumentary system of mammals is quite similar with that of the birds, and actually there are some differences are also present. Histologically, the integumentary system is quite similar with of mammals and birds, but there lies some differences. The first difference is that the basic characteristics of the bird integumentary system is the bird have the feathers, and that feather is very much unique and a very silent and unique evolutionary character that is present in case of birds, and that feather, the presence of feather is a definable character of the birds. But in case of mammals, the basic character that is present in mammalian skin or mammalian integument system is the presence of what? The hair. Exactly, the hair. The hair is an unique character, or you can say a very much salient character which is present in case of mammals. This is one type of, or one case of difference. And another one is the presence of the glands. The glands means in case of birds, the bird have a unique gland, one definable gland which is known as the preen gland. I think you have already gone through the video where I have discussed about the preen gland. But in case of mammals, the mammals have two very much you can say that very definable character of gland. One is the mammary gland. Yes, the mammary gland. The mammary gland is the gland, and another gland is the sebaceous gland. So this mammary gland and sebaceous glands are unique gland which is exclusively present in case of mammals. Okay, so this is the introduction of that particular lecture, and now I am going to the first slide of that particular topic. Now, the integument of mammals is basically known as the skin. Okay, so the alternative name of the integument of mammals is the skin. Exactly, and the another important character of the skin is that the skin has several kind of derivatives, like the hairs, nails, hoops, claws, horns, and glands. Okay, so you can see here that the particular structure is the skin structure which is present in case of mammals. Okay, now go to the second slide. Yeah, so this is the integument. So integument is basically made up of two different parts. The first part is the cutaneous membrane. The cutaneous membrane means where the skin is made up of three different layers. The first one is the epidermis, which is the superficial epithelium. Second one is the dermis, which is the underlying layer with blood supply. And finally, the hypodermis. The hypodermis is the basal layer, and it is made up of basically the fatty tissues or the adipose tissues. So this layer is sometimes called the adipose layer. Okay, so and the another part is the accessory structures. So accessory structures is basically the hairs, nails and its derivatives, horns and its derivatives, and several exocrine glands. I hope you know about the exocrine glands. So the exocrine glands is a gland which do not have, which have actually some ducts. So it is a duct gland. So this gland obviously have ducts, and that is why it is known as the exocrine glands. So this accessory structure is sometimes also known as the skin derivatives. Okay, so now go to the next slide. Yeah, now come to the functions of the integumentary systems. So the functions is very much important. Why? Because it gives the body the protection, the first line of defense from different external sources. For example, the bacteria, viruses. So these two are the basic foreign agents which has to be inhibited to enter into the body. That is why the protection by the skin is very much important. So now the second one is that the protection of the skin from different external radiations. For example, the UV radiations, which is continuously comes from the sun, and the body has to prevent that kind of radiation. Otherwise, what will happen? The skin cells going to mutate, and finally that mutated cell is going to be a cancerous cell. Okay, so this type of protection is very much important for the survival of the organisms. So the protection is very much important. So the first function of the integumentary system, or you can say the skin, is to give the organisms, or you can say us, the protection. Okay, so now the next function is the production of the vitamin D. Yeah, the production of vitamin D. So why do our cells, or why do our body or organisms need to produce the vitamin D? Because this vitamin D is very much important for the calcium absorption by the bones. Exactly. So the one basic hormone which is known as the calcitonin, that calcitonin is responsible for the absorption of the calcium from the blood to the bone, and that absorption is important, and that particularly that absorption is responsible, and that absorption is, you can say, is happened because of the presence of the vitamin D. So now how the vitamin D is produced, that is the question. So to answer the question, you have to go beyond that particular topic, beyond this histological learning. So basically the sun is responsible for the vitamin D production. But how? The sunlight contain different types of radiations. For example, the UV or ultraviolet radiation. Importantly, the nanometer, the 290 and 315, that particular range of sunlight or wavelength of light or UV light is known as UVB or ultraviolet B radiation. And that ultraviolet B radiation is ultimately absorbed by the skin, and the skin is responsible to convert the 7-dehydrocholesterol, or which is also known as 7-DHC or 7-dehydrocholesterol, and the skin is responsible to convert that 7-DHC into vitamin D. Okay, and that vitamin D is mostly produced in the keratinocytes of the epidermis, mostly the stratum basilae and the stratum spinosum. That two layers, that stratum basilae and stratum spinosum, is responsible for the conversion of 7-DHC into vitamin D. Okay, so this is the second function. Now the third function is the sensation. Exactly. Why sensation is important? Because the organism has to sense several dangerous circumstances in the outside of the environment. So in our skin contains several types of sensory receptors, and those receptors is particularly important to receive the dangerous signals from the environment, and gives our brain the sensation that the environment is full of dangerous situations. So our body cope up with that particular situations, and we sense that dangerous situation or circumstances. So this is the third function. Now go to the fourth function. That fourth function is very much important. Why? Because the fourth function of the skin is to regulate the body temperature. Now the question is how? I have already mentioned in my previous lecture that our body contain the blood vessels. So in very warm condition, or when you travel into some hot weather, our dermal blood vessels get dilated. So this dilation of the blood vessel is known as the vasodilation. I repeat again, the dilation of the blood vessel is known as vasodilation. So due to this vasodilation, the vessels of our skin carry more blood to the surface, and That is why, and resultantly the heat is dissipated to the outside, so our body get cooled. So now in this second case where there is very cold temperature, the same thing happens but in different vice, or in different in that case the dermal blood vessels get constricted, and that particular constriction of the dermal blood vessel is known as vasoconstriction. So during vasoconstriction the heat cannot escape from the body to the outside, and our body retain the produced heat, and finally our body retain its heat and it's become hot or warm. So this is how the body regulates its temperature. Now finally the last function is the excretion. We all know, we all quite experienced with the sweating during the summer season. So during summer season our body excrete several types of waste products through perspiration. Okay, so these are the different functions of the integumentary system of mammals. Now, how the skin looks like, or what is the structure of the skin? So basically the skin is made up of or consists of three different layers that I have already mentioned. The first one is the epidermis, the epithelial tissue. The second one is the dermis, or dense connective tissue, or which is totally irregular in structure. And finally the hypodermis, or which is known as the subcutaneous tissue. And the subcutaneous tissue is also known as the adipose tissue, because this tissue is totally made up of fatty layer. Okay, so that is why this tissue is known as adipose tissue. Okay, so now I go to the structure of the skin, the diagrammatic presentation of the skin. So you can see here that particular structure is made up of different layer. So the epidermis is very small or very thin layer present in the outside of the skin, and below that the dermis is present, which is a very thick layer, and finally the hypodermis or subcutaneous layer. Okay, now little bit of the characteristics of the epidermis. So epidermis is basically made up of a stratified squamous epithelium. Why that particular layer is known as squamous or stratified? Because that particular layer is tightly packed, dense, and this particular layer is very protective, and obviously it helps or to help the skin to inhibit or to retain the normal wear and tearing function. Now the different nutrients and oxygens are diffused from the capillaries through that particular epidermis, and the surface keratinized cells are continuously wear off or extrafoliated and replaced by the cells arising from the deepest layer of the epidermis. Okay, so basically that epidermis is made up of different types of cells. Okay, the first one is the keratinocytes. The keratinocytes is basically made up of a protein which is known as the keratin. So keratin is basically a fibrous kinds of protein, and 90% of the epidermis cell is made up of that particular keratinocytes. Okay, so keratinocyte, the production of the particular protein or keratin is basically formed by different molecules or different kinds of hormones or vitamins. For example, calcium, cathepsin, vitamin D3, vitamin A, epidermal growth factor, keratinocyte development is needed that kind of molecules. Okay, so this cathepsin, vitamin A, vitamin D3 is responsible for the production of that particular keratin protein, which is present in large amount in keratinocytes. Now the keratinocytes are the most abundant cells in the epidermis, and from that keratinized or dead horny layer is formed on the outer side of the epidermis because of that particular keratinocytes. Now the second cell, which is known as the Langerhans cells. So Langerhans cells is basically made up of a protein which is known as Langerhans protein. Okay, so that Langerhans cells basically is immune cells, or it is a kind of dendritic cell which is present in the epidermis, and it helps to mediate several types of immune responses from the external foreign agents. Okay. Next one is the Merkel cells. That Merkel cells is also known as Ranvier cells, and one of the scientists who known as the Friedrich Merkel actually named or discovered that particular cells, and that is how that particular cells is named as Merkel cells. So now that particular Merkel cells is responsible for tactile stimulation. So tactile stimulation means that particular Merkel cells is also acts as a mechanoreceptor cells. I am repeat again, because of that tactile stimulatory function, that Merkel cell is also known as mechanoreceptor cells. Okay, and that particular Merkel cell is very abundantly present in the fingertips of our fingers. Okay, so now go to the next cell. Finally, the chromophores or chromatophores. That chromatophores, as you have already get the hints from the name of that particular cell, that cells is loaded with several types of color pigments, or that color pigments is responsible to give the skin a particular skinny color, and the skinny color is responsible for the presence of a particular granular pigment which is known as the melanin. That particular melanin is passed directly to the surface of the epithelial layer and gives the skin a particular color, and also that particular melanin is abundantly present in the shaft of the hair. Okay. So now this is the histological section or a diagrammatic representation of different layers and different cells present in the epidermis. For example, you can see here that particular Merkel cell is present in the stratum bacilli, and it is a tactile receptor is attached with that particular Merkel cell, and you can see the presence of the Langerhans cells. Basically, this Langerhans cell is present in the stratum spinosum, and you can see the stratum spinosum, stratum lucidum, all are filled with a particular cell which is known as the keratinocytes. Okay, now go to the structure of the epidermis. So basically, the epidermis is made up of 5 different layers. Okay, from the base, it is situated upside of the epidermis, and the layers are known as stratum bacilli, stratum spinosum, stratum granulosum, lucidum, and corneum. So when these 5 layers is present in a epidermis, that particular epidermis is known as the thick skin. Okay, which covers mostly the palms of the hands and the soles of the feet. Okay, so because of due to the presence of the 5 different layers of the epidermis, that epidermis is known as the thick skin. But in case of thin skin, which covers most of the body, where the 4 different keratinocyte epithelial layer is present, basically or importantly, the stratum lucidum is vanished from the thin skin. So the thin skin is basically made up of 4 layers except the stratum lucidum. Okay, so this is the histological section, or you can say the longitudinal section of the epidermis. You can see that this is the stratum corneum, which is totally squamous, the stratum lucidum is thin layer, then stratum granulosum, then stratum spinosum, and finally the stratum bacilli. That stratum bacilli is attached with the dermis. Okay, so now the basal layer of the epidermis, or the first layer of the epidermis, which is known as the stratum basilae. That stratum basilae is basically attached with the basement membrane by hemidesmosomes. So hemidesmosomes, you can remember from your last semester where you have learned about the cell adhesion molecules. Okay, so hemidesmosomes are basically some kind of cell adhesion molecules which help for the attachment of two different types of cells. Okay, so now the stratum basilae is actually form a connecting link or connection between the epidermis and the dermis. So that forms a strong bond between the epidermis and the dermis. Now the epidermis, the epidermal layer is actually do some kind of invagination, and that invagination is actually responsible for the formation of several kind of ridges. Okay, so that ridges is basically known as the epidermal ridges. Contrastingly, the dermal layer or the dermis forms some kind of pimples which is known as the dermal papillae or tiny mounds. That dermal papillae is actually help to increase the surface area of the basement membrane. So now the question lies: why do the dermis actually increase its surface area? So the answer is to after increasing the surface area, the dermis can easily diffuse some important molecules, for example the oxygens, some nutrients easily from the dermis to epidermis and vice versa. Okay, and next function is of the dermal papillae is that it strengthens the attachment between the dermis and epidermis. So basically the epidermal ridges and dermal papillae both are make a strong bond so that the connection become very much strong. Okay, now finally the stratum basilae has many basal cells or germinative cells, which is a kind of stem cells which undergo mitosis to produce the constant supply of keratinocytes, which is needed for the replacement of the dead and dying cells, which is continuously wear off or extrafoliated from the surface layers. So basically the stratum basilae is always lies in a dynamic equilibrium. Why dynamic equilibrium? Because that stratum basilae is continuously produce the cells and replenish the cells which is continuously extrafoliated from the surface layers. Okay, now to the next slide. Yeah, that picture you can see here that this is the epidermis and this is the epidermal ridges. Okay, and this is the dermis and this is the dermal papillae. So you can see here that epidermal ridges and dermal papillae form a strong connection or strong connection with each other to form a very strengthened bond, and which is responsible for the strong attachment of the epidermis and the dermis. And very importantly, that epidermal ridges is responsible for the development of the fingerprints in our fingers. Okay, in the top of our fingers, you can see that these are all the epidermal ridges. Okay, so the study of fingerprints is known as dermatoglyphics. Okay, I repeat again, the study of fingerprints, the scientific study of fingerprints is known as dermatoglyphics. Okay, now the stratum spinosum, this layer is very much spiny in appearance. So this is actually produced by the mitosis of the cells present in the stratum basilae. So basically this layer is made up of 8 to 10 layer of keratinocytes, which is bounded by desmosomes. So desmosomes, in this case the desmosomes are again the cell adhesion molecules which help to link or to link or for the formation of a strong bond between same kind of cells, and that desmosomes is actually lies in this particular layer for the formation of that stratum spinosum layer or the second layer. Now because of the presence of these desmosomes, that particular layer looks in a spiny appearance, and that is why that layer is known as stratum spinosum. Now, that particular layer is continuously divided and increasing the thickness of the epithelium. Okay, so, and most importantly, that particular stratum spinosum contain some kind of dendritic cells, which is known as the Langerhans cells, which is active in case of immune response. So these are actually immune cells which is present. So the Langerhans cells are basically the immune cells which is present in the stratum spinosum. Now the stratum granulosum, which is the grainy layer. That means it contains some kind of granules. So basically that stratum granulosum is stop dividing and start producing two different kind of cells or two different kinds of structures. One is the keratinocytes. I have already mentioned about the keratinocytes. It is a tough cell which contains some kind of protein, the keratin protein. Okay, and the keratinocytes is linked with each other by a cementing substance which is produced by the cells present in the stratum granulosum. That cementing substance is known as the keratohyalin. Okay, so that keratohyalin, it's a dense granular structure which is responsible for the linkage between the keratinocytes present in the stratum granulosum. Okay, so keratohyalin is a cross-linking agent. Sometimes questions come in your exam that what is keratinohyalin? The answer will be the keratohyalin is a cross-linking agent which cross-link the keratin fibers present in the stratum granulosum. Okay. Now the stratum lucidum. So it is a very clear layer found only in case of the thick skin which is present in our soles and fingers, and it is totally made up of dying keratinocytes, contain lots of keratin and covers mostly the stratum granulosum. Finally, the stratum corneum. It is a horny layer and exposed to the surface, and it is made up of about 15 to 30 layers of keratinized cells. Importantly, this layer is very much water resistant, and that is why our whole body is become waterproof. Now it shed and replaced by every 2 weeks. Okay. Now the second layer, which is the dermis. The dermis is basically located between the epidermis and the hypodermis below, and the dermis is basically present in the dermis. Several types of structures are present. For example, there are sweat glands, hair follicles, vessels, all are present in this dermis, and the dermis is associated with the nerves, blood vessels, muscles, all are associated with that particular dermal layer. This dermis is made up of two different layers. One is the outer layer is known as the papillary layer, and the inner layer or the deeper layer is known as the reticular layer. The two layers has different characteristics. The papillary layer, which is the outermost layer, is mostly composed of areolar tissues and contain the smaller capillaries, lymphatics and sensory neurons. And the dermal papilla, which is projecting between the epidermal ridges, is basically present in this particular papillary layer. And in contrastingly, in case of the reticular layer, which is made up of dense irregular connective tissues and contain the larger blood vessels, lymphatic vessels and nerve fibers. And importantly, that layer contains a vast amount of collagen and elastic fibers or elastins. Okay. Now you can see here, so this is the schematic diagram of the dermis, and this is the papillary layer and this is the reticular layer. So you can see here that the small blood vessels, nerve endings, lymphatics, all are present in the papillary layer, but not very abundantly. But in case of reticular layer, the vast blood vessels or very thick blood vessels, lymphatics and nerve endings are present in the reticular layer. Okay, so the papillary layer and reticular layer are basically two layers which contain the most of the muscles, most of the you can say the blood vessels, lymphatics and nerve endings. Okay, and you can see that the hair is also originated from that particular layer. Okay, and these are the dermal papillae, and that is why this layer is also known as the papillary layer, because of the presence of the dermal papillae. And these are the epidermal ridges. Okay, now the hypodermis, or the last layer, or which is also known as the subcutaneous layer. So this layer has several characteristics. So it is basically stabilizes the skin, it allows the separate movement, made the elastic areolar adipose tissues, and that is why it is also known as, because of the presence of that adipose tissue, this layer is also known as the adipose layer. And it deposits subcutaneous fat, and it's connected to the reticular layer of the integument by connective tissue fibers. And the distribution pattern of this layer is determined by the hormones. How? Because you can see in case of the females, that particular fatty layer is very thick. Okay, and that thick fatty layer is because of the presence of the female sex hormones, for example the estrogens and progesterones. Okay, in contrast to males, that layer is very much reduced because of the presence of the testosterone, or the absence of the particular two hormones, estrogens and progesterones. Okay, and that layer basically reduced, sometimes this layer is reduced by some cosmetic surgery, which is known as lipoplasty or liposuction. Okay. Okay, so now the glands. These glands are the most important structure present in the integumentary system. So by definition, the gland is a group of cells in an animal body that synthesizes some substances for release into the bloodstream, which is known as the endocrine glands, or sometimes the secretions are present in the cavities inside the body, or it's released to the outer surface by some pores, and that particular gland is known as the exocrine glands. So now we will discuss about the exocrine glands present in our, or in mammalian integumentary systems. Basically, three types of exocrine glands are found. Number one, the sudorific gland or sweat gland. Number two, the sebaceous gland. And number three, the mammalian gland or mammary gland. So now that particular mammary gland is very much important. Why? Because that particular group of organisms or mammals is recognized by the presence of the mammary gland, and that mammary gland is a unique evolutionary character present in the mammals. Okay, so now discuss about that particular glands. Firstly, the sweat glands. So basically the sweat glands are long and coiled and tubular type of glands, and if you see the picture, you can see that the lower part of the sweat gland is present in the reticular layer of the dermis. This is the reticular layer, and this is the papillary layer of the dermis. I have already mentioned that there are two types of layers are found in the dermis. One is reticular and another one is papillary. So basically that sweat gland is present in the reticular layer of the dermis, and the ducts which comes out from the gland is traveled throughout the reticular layer, then finally the papillary layer, and finally to the surface after traveling the epidermis and released by a pore. Okay, so now the function of the sweat gland. The sweat glands mostly function by secretion a salty liquid or salty substance which is known as the sweat. So the sweat basically functions to remove the nitrogenous wastes and helps to regulate the body temperature. So now most interestingly, in human body or in mammals, there are basically two types of sweats are found. One is type of sweat is the thin sweat, which is not associated with the hair follicles and functions before the puberty, and it produces a secretion which regulate the body temperature. Okay, this is one type, and another type is the viscous sweat or thick sweat, which is not associated with the hair follicles and begins functioning at the time of puberty, and it is mostly responsible for the body odor. So basically two basic type of sweat are found. One is the thin sweat, another one is the viscous sweat. So now basically the sweat glands are not found in all the mammalian groups, and their distributions varies. How? Like humans and chimpanzees, they have the mostly the sweat glands are found mostly in the palms, soles and armpits. Okay, but in case of duckbill platypus, the sweat glands are limited to the snout. And in case of cat, dogs and rats, the paws and feet are the location where the sweat glands are actually found. But in case of rabbits, the sweat glands are most abundantly found in the surrounding the lips. But in case of hippopotamus, it is mostly present in case of pinna. But in case of cetacea and sirenia, the sweat gland is totally absent. Okay. Now, the scent gland. What is the scent gland? So, scent gland basically are the modification of the sweat gland. So, scent gland is a modified sweat gland. Sometimes the questions come that in your exam, that's what is scent gland. So, your answer will be the scent gland are a modified sweat gland. Okay. So, the functions of the scent gland is mostly in case of social communication, sometimes the territory marking and kin recognition. Kin recognition means where some organism recognize its own type of organism or its own types. That means its kins or its relatives. So that scent glands are mostly responsible for relative recognition in some animals. For example, the deer and rabbits, they have the chin is the location or the region where mostly the scent glands are found. In case of antelopes and bats, the face, carnivores, the chest and arms, musk deer, it is belly, and camels, it is the back. Okay. Now another important gland, which is known as the sebaceous gland. So basically sebaceous glands are alveolar in nature and associated with the hair follicles, and the secretion of the sebaceous gland is known as the sebum, and the function of that particular sebum is to lubricate the body surface. Okay. So these glands are totally absent in case of pangolins and cetacea and sirenia, but the sebaceous glands are mostly absent in palms and hands and soles of the feet, but they are present without associated hair at the angle of the mouth, on the penis, near the vagina and next to the mammary nipples. Okay. There are some modifications are there also of the sweat glands. For example, the wax gland, which is present in the outer ear canal, and the meibomian glands, which is present in the eyelid, are modified sebaceous glands. Okay. But in case of elephants, this is the organism where the sweat glands and sebaceous glands are entirely absent. Okay. So sometimes some teacher can ask you, or in your viva voce, that name one mammal where the sweat glands and sebaceous glands are absent. Both the glands are absent. So your answer will be the elephants. Okay. So you can see here the structure of the sebaceous gland. There are so many alveolar follicles are present, and the follicles are connected by a duct, and finally the whole gland and the ducts and the whole glands are associated with the hair follicle. Okay. Now finally the most important gland, the mammary gland. The function of the mammary gland is to production of the milk, which is found in all the animals of the mammals. Importantly, that mammary gland is a modification of the sebaceous gland. So here lies another question, that mammary gland is a modified which gland or which gland is modified into mammary gland? The answer will be the sebaceous gland. So basically the gland is made up of a number of lobules. You can see here there is number of lobules are there. Okay, and each lobule is consist of a number of alveoli. So this is one lobule, and these lobules are consist of numerous alveoli. These are the alveoli. This one, all are the alveoli. Okay, which contain the secretory cells. And finally, the small ducts, these are the small ducts are originated or emerged from the alveoli and gradually converge to form a large duct. This is the large duct. Okay, so many such large ducts are united to form a common duct, and finally which opens to the outside through a nipple or teat, in teat or nipple. Okay, so this is the mammary gland. So basically in case of monotremes, you have to remember in the case of monotremes where the nipples and teats are totally absent and the breast do not form, but the milk is released from the ducts into a flattened milk patch or areola on the surface of the skin. Okay, and the front of the infant snout is shaped to fit the surface and permitting the vigorous suckling. Okay, so this is the mammary gland. Okay, so now about the derivatives of the fingers, which is the claws. So claws is basically a ancestral form of digital covering. That means it is an evolutionary character found in mammals. And also the mammalian claw is quite similar with that of the reptiles and birds, and mostly composed of a hard and pointed and narrow curved dorsal plate, which is known as the unguis. You can see here this portion is the unguis. Okay, and a softer ventral plate which is known as the subunguis. You can see this is the subunguis. So this portion is the unguis part or a hard part or dorsal plate kind of thing, which is known as the unguis, and below that one there is a softer ventral plate which is known as subunguis. Okay, so basically the upper surface of the unguis, that means this portion or this portion of the unguis, have a high growth rate, and due to this high growth rate the cells divide more and more, and finally there form a downward curve. This is the downward curve, and this is a unique characteristics of claws. Again, the sides of the claws, that means both sides of the claws were or extrafoliated more quickly than the central portion, so that will produce a sharp pointed edge. Finally, the claws are actually used for the protection of the digits, sometimes for the climbing, sometimes the digging, hanging, and sometimes grasping. But the another function of the claw is that it can kill the prey, to tear apart its fleshes or its muscles. So it also helps during the hunting process. Okay, so now go to the next slide. The nails. We all have nails in our fingers. So basically it is a derivative of the claw and covered most of the dorsal surface of the digit. Importantly, it is the most unique and characterizing feature of the primates. Okay, that means only and one and only the mammals, the primate mammals have the nails just like us. So in case of nails, the unguis is very much wide in structure and thin and less rigid than the subunguis, which is very much reduced. Okay, so in this case the unguis is less rigid than the claws, and the subunguis is very much reduced. You can see here this is the unguis part which is very thin in structure. This is the unguis part which is very thin in structure, and below that there is a subunguis which is very much reduced, and this is the differentiating feature of the nails and the claws. Finally, the nails offer less protection than the claws, but it also helps for the manipulation or precise manipulation of the digits, and that is why we can write, we can draw, or we can press the keyboards, or sometimes our Android phone digits or Android phone keypads. So this is why the nails gives us a precise manipulating structure present in our body. Okay, so beside that one, the nails also protect the mechanical injury of the digits and also help to stabilize the skin at the tips of the fingers and the toes, so that on the opposite side of the skin can establish a secure frictional grip on object grasped. Okay, so now how the nails looks like. So nail is basically composed of a nail bed. You can see here, this is the nail bed. Okay, so this is the visible portion of the nail which covers the nail bed. So nail bed is basically the downward or basal structure which covered by the nails. Okay, so and the pale crescent shaped structure found in the nails is known as the nillula. This is the nillula, which is the crescent shaped structure found in the nail body. And then the sides of the nail, this is the sides of the nail. In this case, the epidermis invaginated to form a groove like structure which is known as the lateral nail groove. These are the grooves or lateral nail grooves, okay, which is surrounded by two folds, this one and this one. These folds are known as lateral nail fold. Okay, so this is the basic structure of the nail. Now the hoops. Hoops are basically found in case of ungulates. For example, only the two groups, the artiodactyls and perissodactyls, these are the two groups where the hoops are actually found. Artiodactyls are those organisms who have whose body weight is actually lies on two fingers, the third finger and the fourth finger. Okay, and that is why the artiodactyls are also known as even-toed animals. Contrastingly, the perissodactyls, who are known as the odd-toed animals or odd-toed organisms. In that case, the total body weight lies on only on the third finger. Okay, so this is the distinction between the artiodactyls and perissodactyls. For example, artiodactyls are the organisms like the cattles, horses, sheep, deers. These are all comes under the artiodactyls group. But the perissodactyls are like rhinoceros, horses. These are comes under the perissodactyls groups. Okay, so in these two groups there is a U-shaped or V-shaped unguis is enclosed both the end of the digit and the subunguis. You can see here, this is the unguis or this is the unguis part. Okay, so this unguis mostly cover the subunguis part and the other parts of the hoof, and the softer subunguis touches the ground and wears away more quickly. So this portion, the subunguis portion, is actually the portion which touch the ground, and this portion is wears off more quickly than the unguis part. And the subunguis, touches a softer horny pad. This subunguis touches a softer horny pad behind the subunguis. So this portion is known as frog or cuneus. Okay, so in case of ungulates, normally only the hoof portion, that means this portion actually touches the ground, but not the cuneus or the frog part. Okay, next one. So this is basically the diagrammatic representation of the hoof of the horses. So we can see here, this is the ventral portion of the hoof, and this is the cuneus part or the frog. This part never touches the ground, rather only this portion or the hoof is actually the part which touch the ground, and this portion wears off more quickly. You can see here also, this is the subanguis part, this is the hoof part, and this is the frog or cuneus. Okay. Now the most interesting structure of the mammals, that means the hairs. It is totally unique to mammals. So this hair is a definable evolutionary character present in the mammals. So now the question comes, how the hair developed? So basically in this case, the epidermis, this is the epidermis, okay, the green portion is the epidermis, and the red cells or red portion are the dermis. So in this case, the epidermis invaginated into a small portion to form a follicle or placoid. Okay, so this portion is actually known as the dermal condensate. Dermal condensate. So the epidermis invaginated to form a dermal condensate, which is known as the placoid. Okay, and the deepest point of the follicle, the dermis pushes back and form a cup-shaped structure which is known as the dermal papilla. Okay, you can see here the dermis pushes back towards the epidermis and form a papilla-like structure, pimple-like structure, which is known as the dermal papilla. Okay, so now finally the epidermal cells on the top of the papilla multiplied and grow continuously and pushed towards the surface and finally it keratinized and formed the hair, and this is the final structure of the hair, and this portion is the hair root, and this is the shaft of the hair. Okay, and you can see here the melanocytes, which is the pigmented cells, also found in the hair root, and then the base of the hair, where the hair just emerged from the skin, here lies a sebaceous gland, which gives the hair a oily or lubricating structure, and that is why the hair looks shiny, because of that sebaceous secretion or sebum secretion. Okay. Now the structure of the hair. Basically the hair is composed of two different parts. One is the hair root, another one is hair shaft. The hair root is the root portion or the lower portion of the hair, and this portion is the hair root, which is attached with the integument. But the hair shaft is the upper part which emerged from the integument, and it is not attached with the integument. So this is the hair root and this is the hair shaft. You can see this presence of the sebaceous gland here, and the erector pili muscle. This muscle is important to move the hair structure, and this is the boundary of the hair shaft and the hair root. You can see here that there were several connective tissue sheaths are present. The hair bulb, hair papillae is there. I have already mentioned this is the hair papillae. Okay. Now go to the ultrastructure or cross-sectional view of the hair. So basically, if you cross-section the hair, you will find there are three basic structure is present. One is the medulla, the most inner part, then the cortex, the middle layer, and finally the cuticle. Okay, so medulla is basically the very central part of the hair shaft or core region, which is composed of mostly dead cells and soft keratinized cells, and the cells are loosely packed, and there is the spaces is filled with air, and that is why the hair become very much flexible. So medulla part is responsible for the flexibility of the hair. Now the cortex. The cortex is basically the middle layer, which is composed of mostly the dead cells, contain the hard keratinized cells which provide the stiffness of the hair, and Mostly the melanins are present in that particular cortex layer. And finally the cuticular part, which is the outermost part of the hair, and it is mostly composed of again dead cells, and this is the part which gives the hair a shiny surface. Now besides these three basic layer, the hair is also surrounded by, the hair shaft is also surrounded by two other layers. Okay, that particular layer do not go beyond the limit of the follicle, and that layer is known as Huxley layer and the Henle layer. This one is the Huxley layer and this one is the Henle layer. So these are the two other layers present in the hairs. So actually the Huxley layer is composed of 2 to 3 cell layer thick and composed of flattened cells, nucleated flattened cells. Okay, and one English biologist whose name was Henry Huxley discovered that particular layer of the hair. Okay, contrastingly, the Huxley layer, which is totally composed of a single, the Henle layer, which is totally composed of a single cell layer and made up of a cuboidal nucleated cells. A German physician and anatomist known as Jacob Henle identified that particular layer in the hair structure. Okay, now to the next slide. So this is the structural presentation of the medulla, cortex and cuticle. This is the medullary part, and surrounding the medullary part there is a cortex layer, and finally this is the cuticular layer, the outermost layer, and this outermost layer is composed of overlapping cells like scales. Okay, now the most important structure of the mammals, the horns and antlers. Again, these two structures are mainly found in artiodactyls and perissodactyls. So I have already given you a brief account of the who are actually the artiodactyls and perissodactyls. So basically, according to its location of the head, on the head and mode of development, the horns and antlers are divided into 5 basic types. For example, the true horn, pronghorn, giraffe horn, hare horn and antlers. So now give a little bit of description of this each structures. Firstly, the true horns. The true horns are basically found in the family of Bovidae, which is present in the order Artiodactyls. For example, the goats, sheep and cattle of the both sexes of that particular goat, sheep and cattle have that particular true horn structure. These true horns are totally unbranched and permanent structure, and that grow throughout the adult life of an animal and never going to shed, and they are mostly composed of a dermal bony core. This is the dermal bony core, which is arised from the frontal bone of the skull and covered by a epidermal keratinized hollow sheath or cap. Okay, so this is the basic structure of the horn. The frontal bone, the true horn is originated from the frontal bone of the brain case, and it is composed of a bony core which is surrounded by a horny or keratinized epidermal covering or cap. Okay, and you can see here, you can notice here, this is the central cavity which is found in the true horn. Now the pronghorns. So pronghorns basically found only in only one species, which is known as the Antilocarpa americana. It is only a living species, only one living species who have that pronghorn structures, and that particular species comes under the family Artiocapridae or Artiodactyls, and is found in the western North America. So anatomically, the pronghorns is exactly same as horns, but it bears about 1 to 3 branches or prongs. So this 1 to 3 these branches are actually known as the prongs. That is why that these horns are known as pronghorns. So these structures are branched obviously and deciduous. Deciduous means they shed annually after each breeding season. That means after completion of the breeding season, that particular pronghorn is shedded, and that permanent bony core becomes base around which a new horn is developed in the following year. So males have the pronghorns, but in case of females it is unbranched and absent. That means only the male Antilocarpa americana have that particular pronghorn structure, and it is shed after each breeding season, and after the completion of the breeding season it shedded, and again the bony core become the base on which a new pronghorn is developed in the following breeding year. Okay. Now the antlers. So antlers is only found in the males of the deer family, and it is fully developed antler is made up of completely of bone. This antler is made up completely of bone. It is also branched and made up of a dense connective tissue and connected basally to the frontal bone of the skull by a bony stump. This bony stump is known as the pedicels. This portion is the pedicels, and the fully developing antler is covered, you can see here, this is the covering, is covered with a skin and short hairs, and this covering of skin is known as the velvet, which carries the blood vessels and nerves and give the nutrition to the growing bone or growing antlers. So when the growth is complete, the blood supply ceases and the velvet is slowing off or wear off, and finally the naked bony antler is exposed. Okay, again after each breeding season the antlers also shed and new antlers developed in the following year. Now a very common question comes in your exams or viva, that what is the difference between the true antlers and true horns. Okay, now the giraffe horn. So giraffe horn is basically unbranched and permanent structures and present in the both sexes of the giraffes, and it is again made up of a bony dermal core projecting from the frontal bone and covered by a velvet, simple velvet, but in this case the velvet never slowing off or never shed. Finally the hair horn or fiber horn. This particular hair horn is the characteristic feature of the rhinoceros group, and it is perched on the nasal bone. So in case of antlers, in case of horns or giraffe horns, all are arised from the frontal bone of the brain case, but in this case in the hair horn or rhinoceros horn it is emerged or perched upon from the nasal bone. Basically the Indian rhino have only a single horn, while the African rhino has two horns, one behind another, and it is totally and made up of a thick, hairy and keratinized epidermal fibers fused together, and importantly these are permanent structure and never shaded. If broken they never grow out. Okay, so this is the part, this is the total part of the integumentary system of mammals. So now I will give you some assignments related to this topic, some questions, some short questions, some broad questions, some descriptive type questions. So you have to solve that particular questions or assignments and give it to me or submit it to me within the stipulated time. So thank you dear students for your attention, and stay home, stay safe and study very tediously. Okay.